Okay. So yeah, student. So here is a question for you. Okay. So today we are going to complete some of the topic of the biomolecules and then we are continuing the human health and disease. Okay. So yeah. So here is a question for you. So this is about enzyme is incorrect. Okay. So here I given some of the statement which is not correct. You have to tell me. Okay. So enzymes are very highly specific. Enzymes are denatured at high temperature and enzymes require optimum pH and temperature for maximum activity. Enzymes are mostly proteins, but some are lipid also. Okay. So can you answer me, student? I will run the poll. Okay. Yeah, you can answer me in the poll. Yeah. Before launching only, you are telling that yes. The correct answer is yes. Option D, right? Yes. The enzymes are mostly protein, but some are lipids also. No, no enzyme is lipid, right? Some enzymes are RNA, right? RNA. They are made up of RNA, so that we can call it as the ribozyme, right? Ribozyme. So that's why option D is the correct answer. Yes, the enzymes are very highly specific. The enzymes are denatured at high temperatures. Enzymes pH and temperature, right? Yes. So the wrong option here or correct wrong statement or correct option is lipid. The enzymes are mostly proteins, but some are lipids. Okay. So student, today we are going to talk about the inhibition of the enzyme activity. Okay. So what do you mean inhibition is correctly? Okay. So see here. So reduction, reduction or stoppage of activity of the enzyme due to the presence of the adverse condition or chemicals is called as the inhibition. So when we are talking about the enzyme optimum temperature and optimum pH, so we have studied about if we increase the temperature or if we decrease the temperature, the enzyme activity is going to be low, right? It is going to be lower the enzyme activity, right? But here in the inhibition, so we are talking about the chemicals, okay? These chemicals will particularly come to the active site of the enzyme, okay? So about this chemical, we are going to study here, okay? Let us start. So student, when it comes to the inhibition, there are two types. One we can be reversible, okay? One can be reversible reaction, and another one is irreversible inhibition, okay? So see here, reversible inhibition. What will happen in the reversible inhibition? So 1 minute. Okay. So see here, it is a type of the inhibition where active site of the enzyme is blocked by the other chemicals. Okay, other chemical substances. Once the chemical substances was removed from the active site, the enzyme is become active. So what happened directly in the reversible inhibition? Okay, for example, so you usually come and sit in a one bench, right? Daily, right? What happened one day? Your friend come and sat here. Your friend, okay, your friend come and sat here. What you will do now? So you'll tell to your friend that this is my place. You go to the another bench, okay? So your friend will get get from that bench and she will sit in her bench, right? So this is what we call it as reversible inhibition. So see here, the where you will sit that is the active site. Okay. So that is the you are the substrate and the here the enzyme is you can remember that bench is the enzyme. Okay. So it is having the active site where you will come and sit. Okay. So now what will happen? Whenever the other substances other than the substrate come and bind to this active site, that is what we will call it as the inhibition. Okay. So see here what happened in the inhibition. This is the enzyme. Okay, this is the enzyme. So this is the active site of the enzyme. Okay. So now what will happen? This is the substrate. Okay, this substrate is going to come and bind to this active site, right? Then only it will convert that into the product. Then only it will convert into the product side. But what happening here is so another substance which is similar to the substrate. Okay, it is similar, very similar to the substrate. See here, similar to the substrate, it come and bind to the active site. So now what? So the active site become blocked. Okay. So now substrate cannot come and bind to this. Okay. So this is what we'll call it as the reversible inhibition. What is the mean by reversible mean? We can make the enzyme active after removing this inhibitor. Okay, so if I remove this inhibitor, now my enzyme become active. Yes, my enzyme now become active, right? Now again it can bind to the substrate itself only, right? So this is what we will call it as a reversible inhibition. The reversible inhibition means the enzyme activity can be restored after the removal of the inhibitor. Okay, so that is what we will call it as a reversible inhibition. So what happened in the irreversible inhibition? Is it okay? Okay, fine. I will move once again. One minute. You can tell me whether it is showing again or not. Okay. Thank you so much. Can anyone tell me now it is moving or not? Can you tell me, student, whether it is moving or not now? Yes. Okay. Okay. Thank you so much. Okay. See here now what happened. So right. Okay. So now you know about what is the reversible inhibition. Reversible inhibition, the inhibitor will bind to the active site when it is removed from the active site. Now enzyme is active only. Okay. So coming to the irreversible inhibition, what happens? So the inhibitor will come and bind to the active site. Okay. But the inhibitor cannot able to remove, or the inhibitor can be removed, but it damages the enzyme. So it cannot bind to the substrate again. It will damage the enzyme active site. Okay. The substrate cannot bind again to the that enzyme active site. Okay, so the damage created by the inhibitor is not be cured. Okay, so that is what we will call it as the irreversible. Okay, see here, irreversible inhibition is a type of the inhibition where active site of the enzyme is permanently damaged. Okay, for example, like this here, the enzyme active site is like this. Okay, so now inhibitor will come and bind to this, and what it will do, it will damage the enzyme active site like this. See here, like this. This is the substrate, right? So how can this substrate can bind to this? Is it possible to bind to this active site now? No, right? So this is how the inhibitor will damage. Okay, inhibitor will damage the active site so that the substrate cannot bind again to the active site of the enzyme. Okay, so that we call it irreversible inhibition. I think you understood. If you didn't understood me, kindly tell me once again. I will repeat. Okay, so let us go to the next. So what is a competitive inhibition? So obviously, if you are writing for the competitive exam, so if all are your 12th standard students, you are competing for the same NEET exam, right? Yes. So here also same thing will happen. So in the competitive inhibition, what happened in the competitive inhibition? Here, the substrate and inhibitor it looks same, okay? Same. It looks same, okay? This is the substrate, and here this is the inhibitor. Okay, it is the same. So it both should have to go and bind to this active site of the enzyme. Active site of the enzyme. See here, whenever this first one is the substrate. Okay, substrate. This second one I'm writing. So this is inhibitor. Inhibitor. Okay. So whenever the substrate will come and bind, so then it is converted into the product. Right? Obviously, the substrate bind means it is going to convert into the product. But here what happens? When the inhibitor will come and bind, so it is going to be block. Okay, it is going to be block the active site. Block the active site. Okay. So now what happens is the inhibitor come around and it sat on this active site. So now the substrate do not have the place to sit in the active site, right? So that is what we will call it as the competitive inhibition. Okay, both the substrate and also the inhibitor is competing for the same active site. Okay, so see here, where the substance similar in the structure to the substrate competes with the substrate for the active site, but could not undergo the chemical reaction. But it cannot convert the substrate into the product, right? Could not undergo any chemical reaction when inhibitor bind to the active site. Okay, so this is what happened in the competitive inhibition. So students, if the any substance that is similar to the substrate, Okay, the any substance similar to the substrate, that we will call it as the substrate analog. Okay, it is similar to the substrate, right? So that's why we'll call it as substrate analog. So what is the substrate analog? The substance which is similar in the structure of the substrate. For example, I will tell you. Do you remember what is a KCA cycle, student? Tricarboxylic acid cycle. So in that you have studied out the enzyme succinate dehydrogenase. Succinate dehydrogenase. Here, the succinate dehydrogenase enzyme, the substrate for the succinate dehydrogenase enzyme is the succinate. Okay, but there is another inhibitor is there, so that is the malonate. Okay, so malonate leads similar to the succinate. Okay, malonate looks similar to the succinate. It will come and bind to the active site of the succinate dehydrogenase enzyme. So, Papa, the succinate dehydrogenase enzyme confused between the succinate and the malonate. Suddenly, what happens? This malonate will come and bind to the succinate dehydrogenase active site. What should it has to do now? So it is blocked, right? The active site of the enzyme was blocked now. The succinate is cannot bind to the succinate dehydrogenase now, right? So that is what happened here. Okay. So that's why we'll call it as the succinate is sorry, malonate is similar to the succinate. When it binds to the active site of the succinate dehydrogenase enzyme, it becomes blocked. The enzyme becomes the block. So now succinate cannot come and bind to the active site of the enzyme, right? So that's why malonate can be called as structural analog of the succinate. Okay, structural analog of succinate. Okay, student, succinate. You have to remember this. Okay, competitive inhibitor example is the this malonate. Okay, analog. So remember here what happened now. We can't reverse this. Okay, the competitive inhibition is a reversible reaction. How can we reverse this? See here. So the enzyme got confusion between the malonate and the succinate now, right? So what I will do now, I will increase the succinate concentration. See here. So this is the succinate. Okay. So here I will write the inhibitor in the yellow color. Okay. So this is the inhibitor. What I will do now, to this I will increase more the concentration of the substrate. Here, more the concentration of the substrate I will add now. Okay. So now the enzyme is blocked by the inhibitor, right? So this inhibitor is binded here, right? So what I am doing now, then adding more and more substrate now. Okay. So now the enzyme recognize this and it also knows, God, I bind it to the inhibitor now. Okay. So now it will be removed. Okay. The inhibitor will be removed from the active site and the substrate is going to be binded to the active site of the enzyme. So when you increase the active, sorry, 1 minute. See here, my enzyme active site only damaged here. I will correct this. Okay. Yes. See here. So now what happened? So this correctly now come and bind to the substrate, right? Now after increasing the substrate concentration, my substrate come and bind to this active site of the enzyme. So now this substrate can be converted into the product, right, student? So this is what happened in the competitive inhibition. You have to remember here, the competitive inhibition is a reversible reaction. So when we increase the substrate concentration, so the enzyme activity is going to be reversible. Okay, it is going to be reversible here. Okay, so the example is inhibition of the succinate dehydrogenase enzyme by the malonate. Malonate is the analog of succinate. Okay, fine. So let us talk about the non-competitive inhibition. Okay, so it is the non-competitive, or we'll call it as allosteric. Okay, non-competitive. So what happened in the non-competitive? See here. So the substrate and the inhibitor, there in the competitive inhibition, they are similar in structure, right? But here in the non-competitive inhibition, so the substance is only different structure and inhibitor is only the different structure. Okay, the substrate is only the different structure and the inhibitor is only the different structure. And they both are not competing for the active site. Okay, so the substrate will bind in the active site only, but the inhibitor is going to bind in the allosteric site. Okay, see here, to where the inhibitor, see here, inhibitor is going to bind where, student? In the allosteric site, allosteric site, right, where the substance is going to be bind in the active site, right? So that's why we'll call it as a non-competitive, okay? The structure is also different and both of them is not competing for the active site also, right? So yeah, inhibitor is telling, aram se you can bind, the inhibitor is aram se going to bind to the allosteric site. So here the substrate is going to bind in the active site only. Then what's happening in the inhibition now? See here. See here, student, carefully. So carefully see this diagram. So this animation, the inhibitor, before binding of the inhibitor, see what happens. The structure is same like this V structure, right? V structure. The substrate can bind, come and bind to this active site, right? So here, once the inhibitor bind to the allosteric site, the structure of the active site is going to be differ, right? That it is going to be damaged. See here, the V shape, right? So when see here, now inhibitor will bind, it is going to be change the shape, right? So now this is the shape, right? Yes. So now this substance cannot come and bind tightly to the active site, right? It cannot come and bind tightly to the active site. So that's the reason. So this is what we call it the inhibition, non-competitive inhibition. After the binding of the inhibitor to the enzyme, it is going to modify or it is going to change the active site, okay, active site of the substrate. So active site of the enzyme, then substrate cannot come and bind to the active site and it cannot convert substrate into the product. Okay, so let us learn what they are given here. Okay, the substance will bind to the enzyme other than the active site. Remember, student. Okay, in the non-competitive inhibition or the allosteric inhibition. Okay, so the substance is going to be bind to the other than the active site. Okay, here is going to modify the enzyme. See here, it is active site is going to be modified, right? Yes, so that the substrate cannot bind to the enzyme and catalyze the reaction, right? The substrate cannot bind to the enzyme now and cannot catalyze the substrate into the product now. Okay, so this is what happened in the non-competitive inhibition. Okay, so here the end product of the reaction inhibits the enzyme. Okay, so what happened in some of the reactions, like you can take the glycolysis. What is the end product of the reaction? ATP, right? ATP and the pyruvate. So whenever the ATP is rich in the rich, the glycolysis is not going to be happen, right? Because the ATP is going to be act as the allosteric inhibitor. Okay, so ATP, okay, ATP is going to act as the allosteric inhibitor in the allosteric inhibitor, where in the glycolysis, okay, for any of the enzymes. Fine. So what happened here? In the competitive inhibition, what happens, student? So here, the Km of the enzyme, Km of enzyme is going to be increases. Can anyone tell me what happens if the Km of the enzyme is going to be increases? Can anyone tell me? See here, students. When the Km was increases, obviously the affinity of the enzyme, affinity of enzyme for substrate is going to be decreases, right? So that's why it must have the low affinity. Okay, always the enzyme should be have the low affinity for the increased enzyme activity. But here what happening? So here it is having the high affinity. So because of it is having the high Km value. So because of that, it is having the low affinity towards the substrate. Always the enzyme should have the low Km value. Okay, low Km value. So if it is having the high, it is having the low affinity. Okay, so then coming to the next, the Vmax. Okay, Vmax is not changed. Okay, Vmax is not changed. Okay, Vmax is like the constant. Okay, Vmax is not going to be changed, or it is the constant in the competitive inhibition. So what happened in the non-competitive inhibition? See here, Km is not altered. Km is constant here because no one is competing, right? So that's why it is constant. When it comes to the Vmax, obviously it is going to be damage the active site of the enzyme. So the Vmax of the enzyme is going to be decreases. Okay, Vmax is going to be decreases, whereas the Km is constant in the non-competitive inhibition. Okay, student, can you remember like this? It is very easy, right? Game is constant because in the non-competitive innovation, no one is going to be compete, right? So that is the reason. Okay. So in the starting only, I have told you conjugated enzyme and the simple enzymes, right, student? So let us discuss about this thing. So some few things, okay? See here, I have told you that there will be the simple enzyme. So the simple enzyme is made up of only the protein, right? So these are required for the conjugated enzyme. Conjugated enzyme. Okay, see here what happened. So here conjugated enzyme having the two things. One is organic factor and another one is the inorganic factor. See here student, here you have to remember that holo enzyme. What do you mean by holo enzyme? Complete enzyme, right? Holo means complete. See here in the holo enzyme what will happen? So it is having the protein part. So that is the apoprotein, right? Apoprotein part. If it is having the apoprotein part plus coenzyme. Coenzyme. What do you mean by coenzyme? Coenzyme is made up of the organic factors. So it's made up of the organic factor. So this organic factor is made up of the vitamins. Okay, student. So holoenzyme should contain the apoprotein plus coenzyme. Okay. So here the coenzyme is made up of only the organic compounds. So that is the vitamins, NADH, FADH. So these all the things are the protein or the coenzyme. So just remember that vitamin. Okay, vitamin. So then coming to the inorganic cofactors, it may be a metallic ions compound. See here. So here it is having a holo enzyme, right? Holo enzyme, right? So in the holo enzyme, there are two parts, right? One is the apoprotein part. Apoprotein. Here we have to talk about the coenzyme, right? Here we have to talk about the coenzyme. So here, if it is the non-protein, if it is the inorganic, so if it is the inorganic means we will call it as the cofactor. Cofactor. Remember here in the exam, what the organic factor of the holoenzyme means you have to tell that it is the coenzyme. Okay. If they ask you about the inorganic factor means you have to tell that that is the cofactor. Okay. So here you can take in the cofactor it will be a calcium or it may be a magnesium, right? Or it may be a manganese. Okay, student. So this is about the cofactor. Okay. So this is about the things about the this is all about the biomolecules. If you have any questions about the biomolecules, you can ask me or else you can answer to this question. The question is very simple. Okay. The question is the quaternary structure of human hemoglobin. We have studied in the quaternary structure, right? The quaternary structure of the human hemoglobin is best described as. If you know the structure of the hemoglobin, you can answer to this question easily. Okay. Can anyone answer to this question? Can anyone answer to this question? I will launch the poll. You can answer me. You can answer me to this question. JNV Chitradurga. So JNV Chitradurga, you can answer me to this question in the whole in the chat box also. Very easy question. Very easy questions. Very good. Very good. I've got the answer. No, students. No, it is not a correct answer. Okay. I'm going to share the result with you. See here. Yeah, I'm going to share the answer with you. Okay. So everyone chosen the option A. So it is not the correct answer. See here, I will just explain you how. So remember how it is correct. Okay. Very good. Very good, Chitradurga. Yes. Option D is the correct answer. Yes, you came to know this, right? Yes. So when we're talking about the quaternary structure of the hemoglobin is made up of two 2 alpha and 2 beta, right? So then I can call it as the tetramer, right? Tetramer I can call it, right? 2, 2 plus 2, 2 is the 4, right? So that's why we'll call it as a tetramer, right? So then it is made up of the two different subunit, right? Alpha and beta, right? Two different subunit, right? So that's why see here, dimer of the identical subunit, no. Dimer of the different subunit, no. It is a tetramer. So that's why it is not a dimer, right? Tetramer of the four different subunit, no, right? It is a tetramer, correct, but it's not four different subunit. right? So tetramer of two different subunits. Very good, student. Very good. Yes. Okay. So this is about the biomolecules. Let us start our new chapter. Okay. So human health and disease. Okay. So yeah, let us start the chapter, students. Okay. What are the things we are going to study in this chapter? Okay. So I think in some of the schools this chapter has been completed. No problem. So let us start here also. Okay. Yeah. See here, student. So here we are going to study about the common diseases. Okay. So in the common diseases, we are going to study about the bacterial diseases, viral diseases, protozoan, helminthus diseases, these all the things. Okay. And fungal diseases. So this is the thing we are going to study in the common diseases. Then coming to the immunity. Okay. So immunity we are going to study. We are talking about the B cell, T cell, and allergic reaction, antibodies. These all the things we are going to study in the immunity. Then we are going to study about the two diseases. One is the infectious disease and another one is the non-infectious disease. The infectious disease is the AIDS and non-infectious disease is the cancer. Okay. So then we are going to talk about drugs and the alcohol abuse. Okay. So drugs and the alcohol abuse, then adolescence going to study in this chapter. Let us start this chapter. Yes. Okay. Here is a question for you. Can you think, student? I know. So you are brave student. You will tell this answer, right? Yeah. So which one of the following tests is considered as gold standard test for diagnosis of COVID-19? I think everyone knows this answer, right? Before coming to the JNU or before going to the any place, we have to do this test, right? Can anyone answer me? Yeah, in the chat box, you can answer me, student. JNV Chitradurga. Yes, I know you people, you know this answer. Yes, very easy question, right? Yes. So that is real-time RT-PCR. Can you tell me full form of RT-PCR now? Can you tell me full form of RT-PCR? Let us try, you know this, right? RT-PCR, right? Can you tell me full form of RT-PCR? I'm waiting for your answer. JNV. Can you answer me? Yeah. I'm waiting for your answer. I will give a time. Think and tell me. Reverse transcriptase, right? Yes. Reverse transcriptase, right, student? Reverse transcriptase. Why we are using the reverse transcriptase here? Our coronavirus is the RNA, right? It is made up of the RNA. So that's why we are going to do the reverse transcriptase. Okay, we are using the enzyme reverse transcriptase. What do you mean by PCR? Polymerase chain reaction, right? Polymerase chain reaction. Right, student? Yes. So polymerase chain reaction is the correct answer. RT-PCR. Okay. So what do you mean by health correctly? So does that mean that I'm a healthy person? I don't have any disease means, can you tell me as a healthy person? No, right? For example, if you think, so I'm a healthy person, so I'm mentally abnormal. Okay. So I'm behaving very rudely. I am not at all behaving correctly in the social. Okay. I'm not emotionally fit. Can you tell me as the healthy person? No, right? We can't tell that, right? Yes. So whenever we are telling any person as a healthy person, he must have the physical well-being, right? Physically fit, mentally fit, and should be socially well-being and emotionally fit, right? So then only we will tell that it is a healthy person or well-being state. That person is healthy person, right? Yes. So mentally, physically, socially, and the emotionally well-being is called as the health. Okay, student? Fine. So then obviously we are going to talk about the disease. What do you mean by disease, student? For example, if you think, if we are healthy person means our body function is going normally, we are normal, right? We are going to work actively, right? So whenever you are having the headache, a headache, or if you are having any body pain or any fever, how you are going to work? It is not at all possible to work, right? You will take the rest, right? Yes. So what do you mean by disease? The harmful deviation. Okay. So malfunctioning of the body with the specific symptoms, right? If you have a headache means your head is going to be pain, right? So that we will call it as a headache. If you're having the fever means the body temperature is going to be rise. You will give specific symptoms, right? Yes. So that is what we will call it as a disease. Okay. Malfunctioning of the body with the specific symptoms. Okay. So this is okay. So student, when coming to the types of the disease, there will be two types of the disease. Okay. One we will call it as a congenital disease. Okay. Congenital disease. One is the acquired disease. What do you mean by congenital disease? The disease which is acquired from by birth. Okay. By birth, the any person is having the disease means that we will call it as a congenital disease. For example, if you take the chromosomal disorder, Turner syndrome, Klinefelter syndrome, right? So these all are, we can call it as the congenital diseases. Okay. So these diseases we come from the damage of the chromosome itself, right? We can't change that chromosome after the birth, right? So it not be cured, congenital diseases. Okay. So that is what we will call it as a congenital. Congenital is from the by birth. Okay, by birth. Okay. So let us talk about what is the acquired disease. Acquired. So that is we are telling that I have born in my lifetime I am going to acquire a disease. Okay. So that is what we will call it as the acquired disease. So this is in the lifetime acquired. Okay. If I acquired any disease, then I'm going to call it as an acquired disease. For example, if I caught with the cold, okay, that is my acquired, right? I have not got the disease from my parents, right? So that is what I will call it as an acquired disease. If I got the any fever, that is acquired, right? Yes. So this is what we will call it as acquired disease. During my birth, during the after the birth, which are the disease I get, that is we call it as an acquired disease. Okay. So then we will talk about the communicable diseases. Okay. So some diseases are communicable. So how we will communicate, right? In the phone and all the things? Yes. Some disease also communicable, but not through the phone. Okay. So how it is going to be communicable? Let us study it. One we will call it as a contagious disease. Contagious communicable. Contagious means direct. Okay. So this disease is going to be contact directly. Directly means, for example, if any person is having the cold, okay, if you sit with that person, after 3 days, you also going to get the cold, right? So obviously, time also you have seen that if some person is having the corona, so everyone is feared to go in front of that corona people, right? Because it is a contagious, means it is going to be spread through the direct contact, right? And smallpox, chicken pox, if you take these, all the things will come under the direct contact. If that person is having the disease, means if you contact with that person, means you are going to get the disease. Okay, that is what we call it as a contagious disease. Okay, non-contagious means indirect. Okay, indirect disease. Indirect. It is not going to be spread through the by sitting with that person. Okay, by sitting with the diseases person. For example, malaria. Okay, so malaria is going to be transmitted through the female Anopheles mosquito, right? If you sit with the malaria, the person is having the malaria means you are not going to get the malaria, right? The person that malaria female Anopheles mosquito has to become and it should be spread the disease, right? So that is what we will call it as the indirect. Non-contagious means indirect. Contagious means direct. Okay, student, I think you understood this. Okay, so let us talk about the non-communicable disease. It is not at all communicable with the any indirect agent or anything or by sitting with that person. For example, if you take deficiency syndrome, for example, if any person become anemic, okay, anemic means that is because of the deficiency of the proteins, right? If he is not properly taking the dieting food means, then he will become the deficient. He is going to get the disease, right? Anemia. So it is not communicable. Then we come to the degenerative diseases. Any neurodegenerative diseases that happens to that person by sitting with that person, it is not going to be spread to the other person, right? So that is a degenerative. And coming to the addiction, if one person is alcohol addiction, by sitting with that person, you will not get you are not going to be addicted, right? So that is the second thing. And allergic reaction and mental disorders and the cancer. Cancer. Cancer is a non okay, by sitting with the cancer patient or by eating with the cancer patient and by playing with the cancer patient, you are not going to get any cancer. Okay, fine. So this all are comes under the non-communicable diseases. I think you understood this. If you have any questions, you can ask me here, student, or else we'll move to the next slide. Jainvi Chitradurga, do you have any questions here? Can you tell me whether you have any question here, student? Are you understanding what I'm telling? Can you answer me in the chat box? Very good. Okay, thank you, students. Let us go to the next. Fine. So, students, so here there are the two types of the chemicals. Thank you so much. Thank you. Okay. So, student, how is going to be caused? We know that the disease is going to be caused from the bacteria, fungi, and viruses. Is all the thing, right? So, there will be a two agent which will cause the disease. Okay. One we will call it as the exogenous. Okay, 1 minute. Yeah. So one we will call it as the exogenous. Exogenous means what? Outside the body. Outside the body. Outside the body, anything comes and damage the body. Okay. That is what we will call it as the exogenous. Okay. So for example, if you take from the outside, the bacteria came, from the outside, the virus came, right? Or from the outside, any chemical came. Okay. It is causing the disease. So that is what we will call it as the exogenous. Okay. So let us talk about the endogenous. For example, if you think, student, if the sugar is present in our body only, right? If the sugar was increases, you are going to get the disease, right? So what diabetes we will call it as, right? Increases the sugar level in the blood, right? For example, if you got uh, the doctor will tell to check the um, give for the test, right? For example, urine test they will tell, blood test they will tell, right? So for example, you know about the arthritis, right? Arthritis. In the arthritis, what happens? Uric acid concentration is going to be increases. Okay, uric acid, endogenous. So the uric acid is going to be come from the inside the body. Okay, uric acid. So where you are going to get the uric acid from the breakdown of the nucleic acid. Okay, breakdown of nucleic acid. In our body, DNA and RNA is there, right? So we will take the fruits, we will take the plants, right? We will take the vegetables, these all the things, right? So there are the DNA is present in that, right? So that will break down and that will convert into the uric acid. Okay. So that will convert the uric acid. If it is increases in the blood, okay. If it is increases in the blood, that is going to cause the arthritis. Arthritis. Okay. So this is what in the you can see that in the joint there will be a fluid was filled, right? So joint pain, you will get the joint pain, right? That is what we will call it as the arthritis. So that is due to the endogenous chemical which is present in our body only. So that is the uric acid. Okay. So outside the body means exogenous, inside the body means endogenous. Okay. So let us talk about the direct transmission, and some of them will come through the indirect transmission. I have told you. So without which are the things going to come from the direct transmission? What do you mean by actually direct transmission? Let us learn this. Contact with the infected person. Okay. For example, if you take the chicken pox and the smallpox, contacting with that person only you're going to get. And droplet infection, obviously cold with all the things. And contact with the soil, you're going to get the tetanus. Okay, students, tetanus and animal bite, rabies you are going to get, and through the placenta you are going to get the syphilis, these all the things. Okay, and AIDS, HIV, AIDS through the. So this is the thing how the direct infection is going to be come. So infected person, droplet infection, contact with the soil, animal bite, and to the placenta. So see here, I have given that example. Infected with the contact with the infected person, smallpox, chickenpox, and the ringworm. So student, you have to remember these examples also. Okay, so it is very very important. So then coming to the second thing, so droplet infection. If some person is having the cold, if he sneezes, coughing, spitting, so through that also you will get the diseases. Which are the diseases? Tuberculosis, influenza, cold, measles, and the whooping cough. So this all the thing is by the droplet infection. Droplet infection. So they may ask you in the exam like this, okay? So they give the some disease name, and they may ask you. So which one of the disease is going to be caused by the droplet infection? Okay, students, please note down these diseases. Okay, droplet infection, tuberculosis, influenza, measles, and whooping cough. Okay, so see here, tetanus. So, student, you know tetanus injection, TT injection you will take, right? Whenever you are infected with the any rod, iron rod, something, right? Or the small children will eat the soil, right? So there is a Clostridium tetani bacteria will be there. Okay, Clostridium tetani bacteria will be there. Okay, in the iron rods and in the soil. Okay, so because of that, they are going to get the tetanus. Okay, the tetanus is a very dreadful disease. Okay, see here, the person how he became after getting the tetanus. Okay, so this is how the tetanus is going to be. Okay, by the contact with the soil. So that's why if anyone damaged, if anyone is hurted by the iron rod, means they have to get the TT injection, right? So because this bacteria is going to be infected, that is the reason. So we know about the rabies, right? So rabies, so that is infected animal. Not all the dogs biting will cause the rabies. Okay, if that dog was infected with the rabies means if bite you means then only you are getting the rabies. Okay, so and one more thing you have to remember here. Not only that dogs will cause the rabies. Okay, the other mammals also like the rabbit and the cat. This is also going to cause the rabies. Okay. Okay, see here, placenta. So German measles, syphilis, and the HIV. So student, here you have to remember that. So which are the disease will transfer to the placenta? This is also important thing, okay? German measles and HIV and the syphilis. Very, very important. Okay, student. So remember these things. So let us come to the which one of the following disease cannot be communicated through the droplet infection? Okay. Can anyone answer me this question, student? Very easy questions. Now only we studied this, right? Can anyone answer me? Which is the Janvi RR? Welcome to the class. Can you answer me, students? Very easy question. Only we studied this. Can anyone answer me? Option A is influenza. Option B is measles. Option C is HIV/AIDS. Option D is tuberculosis. This cannot be transferred to the droplet infection. Can anyone answer me? Very easy question. Janvi Chitradurga, can you answer me? Students, Janvi Chitradurga. Very good student. Yes, everyone knows this answer, right? Very, very easy question. What, ma'am, you are giving this much of the easy question, right? Yes, very easy question. This is yes. Okay, HIV, AIDS, right? Yes. So these all the things are not going to be. These all the things is going to be from the droplet infection. HIV is through the sexually transmitted disease, right? Okay. So then indirect transmission. Let us talk about the indirect transmission through the intermediate agent. Okay. So student, it may be arthropod vector. For example, you are going to get through the insect and the fly, right? So another thing is the vehicle-borne method. It may be through the air, soil, or through the water, right? So that is the vehicle-borne method. And airborne method, formate-borne method, and last one is the human carrier. What do you mean by formate-borne method? For example, student. So we know that coronavirus. If that person is if the person was touched something, means we should not touch. So because the coronavirus should be there in the pen also or in the book also. The person, if he touch that means the person is infected. If he touch that means there is also the coronavirus will be there, right? So that all the things, that these things used by the patient. So that thing we will call it as the format. Okay, for example, pen or pencil, or we can take the books or any, we can take the, this all the things, some other materials. Okay, this all the things will come under the format method. Okay, so then coming to the human carrier, you all know about the in the textbooks also they are given the typhoid, right? Have you heard of the typhoid Mary, student? So there is a typhoid Mary is the one of the cook. Okay, is a professional cook, and professionally she is a cook. Okay, she used to cook for the so many people. So the people, so she is a carrier of the typhoid. Okay, so the people who all are come to her restaurant, she ate in that hotel. Sorry, the people ate in her hotel. All of them were the typhoid. Okay, so once they went for the checking, so the only thing similar in all of the patient is they used to eat in this typhoid Mary hotel. Okay, so that is the reason for her they are given the name Typhoid Mary, but it's not the Typhoid Mary is not a real name. Her name is Mary Malone. Okay, her name is Mary Malone. Okay, Mary Malone. Mary, sorry, one Mary Malone. Okay, not Typhoid Mary, actually real name. Okay, so Mary Malone is a real name. Okay. So after she is continuously spreading the typhoid disease, so later she got that typhoid Mary. Okay. Typhoid Mary. Okay, student. So she is a human carrier. Okay. In the exam, they ask you, you can tell me typhoid Mary. Okay. Fine. See here. Format born method. So we have our rat. We know that plague is caused by the rat, right? Not rat. In the rat, there will be a rat flea is there. Okay. That rat flea is going to carry the bacteria. Through that, you are going to get the disease. Okay. So that is the plague. Okay. So here the bacterial name is Yersinia pestis. Yersinia pestis. Okay. So here you can see housefly, typhoid, cholera, and dysentery is caused by the housefly. Mosquito is going to cause the malaria, dengue fever, right? So, and the T-say fly is going to sleeping sickness. So here you have to remember, student, trypanosoma, trypanosoma, okay, trypanosoma. It is going to cause the sleeping sickness, okay. So then coming to the leishmania, it is by the sandfly, okay. So let us come to the, so how these agents is going to cause the disease. So one is by the tissue damage, another is by the toxin secretion. See here, so tissue damage. For example, if any person was bited by the, so you know how the rabies is going to be transferred, right? Sorry, how the rabies is going to be damage the tissue. See here, whenever the that rabies dog saliva is having the virus. What is that virus? We will call it as rhinovirus. Sorry, rhabdovirus, rabies virus. Okay. So whenever this dog bited this person, in this saliva, it is having the rhabdovirus. So that is a rabies virus. Okay. See what happens. This virus is going to be replicate here. Okay. One virus is there. What will happen? No, no problem. So first we will replicate. We'll make many numbers. Okay. Then we will transfer. Okay. So see what happened now. It is increases here. Okay. So virus replicates in the muscular near the bite. So next what happens? This virus moves up to the peripheral nervous system. Through the peripheral neuron, here through the neuron ending, it is going to be transferred. Okay, it is going to be transferred through the neurons. Okay, see here through the neuron, it is going to be go to the brain. In the brain, it is going to cause the cerebral damage, cerebral cortex damage, cerebral damage. Okay, student, it is going to cause the cerebral damage. Okay, here virus ascends the spinal cord, it is going to spinal cord, then it is reaches the brain and causes the fatal encephalitis. Then virus enters the salivary gland and the other organs of the victim. So at the last stage, what will happen? So it will replicate in the salivary gland and reaches the salivary gland of the person. So students, so famously we will call it as this rabies as the hydrophobia also. Okay, hydrophobia, hydrophobia. So means the person is infected by the rabies virus having the fear of the water. Okay, why this fear of the water means he has a thirst. Okay, he has a thirst to drink the water, but he could not able to drink the water because it's salivary. What we will call it as the virus is going to multiply here, right? It is damaged the trachea. Okay, trachea. He could not able to drink the water. It is cause of the pain, but he has a thirst. He cannot able to drink that water. Okay, so that's why that we will call it as a hydrophobia. Not only with the water, with the juice also is having the with all the liquid items is having the phobia. Okay, and also is having photophobia. Okay, students. So whenever we infected with the dog with the rabies virus, we have to take the 5 injections. 5 injections. Okay, which is that injection you have to take? You have to tell me in the next class. Okay, student, in the next class, this is the homework for you. You have to tell me which injection will you to the rabies vaccine. What is that vaccine name? You have to tell me. Okay, so see here, there is the two types of the secretion. One we will call it as the exotoxin, and another one is called as the endotoxin. For example, if you take the bacteria, okay, so in the bacteria, when it is enters into the body, okay, enters into the body, it is started releasing the chemicals, right? It is started releasing the chemicals. It is a bacteria, right? If the bacteria started releasing the chemical, that we will call it the exotoxin. It is released from the outside the body, right? Exotoxin. When we're talking about the endotoxin, so student, have you learned about the cell wall of the bacteria, right? So on the cell wall of the bacteria, it is having the polysaccharide coat. Polysaccharide, right? Polysaccharide coat. So this is going to act as an endotoxin. Okay. So this is the exotoxin is released by both gram positive bacteria and gram negative bacteria. But see here, the polysaccharide coat is going to be present only in the gram negative bacteria, right? It is going to present in only gram negative bacteria. Endotoxin is going to be produced by only gram negative bacteria. Okay, this bacteria will go inside and it will burst. Okay, when it burst or when this polysaccharide coat was removed, then it is going to cause the disease. Okay, due to that polysaccharide coat, you are going to get the disease. So that is what we will call it the endotoxin. Why it is called as the endotoxin, student? Because it is a part of the bacterial cell wall, right? Part of the bacterial cell wall. So that's why we call it as the endotoxin. Okay, fine. So it is why we will call it as the endotoxin. Part of bacterial cell wall. Bacterial cell wall. Okay, student. So this is about today. So from tomorrow we'll talk about the bacterial disease and all the things. Okay. So if you have any questions, you can ask me. Otherwise, we will end up the class today. Okay. So student, you understood what are the things we have studied today? We have completed the biomolecules and we have studied about the human health and disease. In that we have talked about the what is a communicable disease and what is an uncommunicable disease, which are the communicable, which are not communicable. Okay, this is name also we can tell. Okay, this is name also we have learned, right? Okay, fine. So do you have any questions? You can ask me. Otherwise, you can leave the class. You can give me the NPS. Okay, your feedback is important. You can give me your feedback, students. Thank you so much, Jainvi Chittadurga. And thank you, Jainvi R. All the best, students, for your studies.